Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about generation of genetically modified organisms, and in the last couple of modules what we have discussed so far, we have discussed about the physiology of host, as well as we have discussed about different types of vector or the transforming agents what you can use to develop the genetically modified organisms in a particular host. In this context, we were discussing about how to produce the protein in different host strains, and in this discussion so far we have discussed about the E. coli as an expression system, and in today's lecture we are going to discuss about the remaining expression system such as yeast as an expression system, insect cell line as an expression system, and lastly we are going to discuss about the mammalian cells as an expression system. So, when you talk about the E. coli as an expression system, we have discussed many aspects. We have discussed about how to clone the protein into the E. coli expression system, what are the different types of host and the strategies which are available for you to produce the protein under the different host systems. And in following this context, now today we are going to start the lecture with the discussion about how to control the protein production in the E. coli expression system. So, there are many factors which are affecting the protein synthesis within the E. coli. One of the major factor which we have discussed in the previous lecture as well is the strength of the promoter itself. So, if you can have the two types of promoter, either the promoter which is very strong promoter, and the strong promoter is going to facilitate the easy binding of the RNA polymerase, and that is how it is going to facilitate the more turnover number of the transcription events, and as a result you are going to have the high level of RNA in the RNA pool, and subsequently as a result you are going to generate the more amount of protein. In addition to that, if you have the if you have the weak promoter, the weak promoter is not going to support the protein production, and as a result you are going to have the low level of protein expressions. Both the strong promoter as well as the weak promoter has its own advantage as well as the disadvantages. The strong promoter gives a very high amount of protein, that is why it is good for the protein production if the protein is small in size and it is easily getting folded. But if it is a big protein and it requires the time for getting folded, then you need to use the weak promoter, which actually going to give the cellular machinery to fold the protein properly. Apart from the promoter as a deciding factor to control the protein production or the efficiency of protein production, you also have to consider about the another parameter which is called as the Shine-Dargano sequences. So, Shine-Dargano sequences actually allows the binding of ribosome machinery to the recombinant DNA, and it actually controls the overall protein production from the particular clone. So, the distance between the Shine-Dargano sequences and the start codon is very important for the efficient transcription as well as efficient translation of that recombinant DNA. Along with that, the secondary structures which are being present in the promoter are also going to affect the efficiency of the protein or efficiency of the gene expression. Apart from that, the growth conditions or the growth media which you are going to use has a very dramatic as well as significant impact on the particular protein productions. For example, you can imagine a protein which you are using and that requires a large quantity of arginine or lysine or glutamine. In those cases, you have to ensure that the media components should have these amino acids or these essential amino acids. So, the growth media, we have already discussed about the growth media which are actually going to be used for growing the bacterial cells. and the growth media has a dramatic effect on the gene expression. Either the media components provide the raw material, this is like what we have discussed so far, whether you are, if suppose your protein requires lysine, arginine and glutamate, then these amino acids should be present or their precursors should be present in the media, or to provide the amino acid for the synthesis of a particular protein. In addition, some of the growth media are very rich in carbon source, and that actually going to generate a large number of cell mass, and once you have the large number of cell mass, subsequently or eventually you are going to have the large number of you are going to have the more production of the particular protein from the cell. So, apart from this, the third factor is the codon usage. You know that the genetic codes which are 64 in number, where you have the 61 codes which are available for the 21 different amino acids, whereas the 3 codes which are UA, UGA and UAG are the stop codons. So, as a result, every organism, so these 61 codons are every organism is using, but every organism has a particular type of preference for a set of genetic codes to express few of the amino acids or a class of amino acids. In those cases, what will happen is that if you are cloning a protein into the E. coli expression system, you have to ensure that the tRNA corresponding to these codons should be available, and this particular type of unique problem is called as the codon usage or the so expressing these sequences requires tRNA to recognize the genetic code, but if the host expression system does not have the tRNA or low level of a particular tRNA, then it will either delay the synthesis or it will stop the synthesis at a particular amino acid. You can imagine a situation that E. coli does not have the tRNA for phenylalanine, which the gene which you have cloned into that particular E. coli is require, then in those cases, as soon as the RNA polymerase or the particular cellular biosynthesis machinery will reach to that particular phenylalanine, it will not going to get the tRNA, and as a result the protein synthesis will stop, or it will going to generate the protein which is of the truncated in nature. So, and this problem can be circumvent if you overexpress these additional tRNAs into the host strain. Either you make the chimeric expression system where you can clone these tRNAs into the chromosome, or you can supply these chromosome gene tRNA genes cloned onto the separate plasmids, and you can transform your plasmid as well as these additional tRNA plasmids. So, if you do so, you could be able to overcome the codon usage problem. So, if that happens, it will actually going to produce less protein or truncated protein. So, if you encounter the codon usage problem, you have to either overexpress these additional tRNAs into the separate plasmid, or you have to generate the chimeric host which will actually going to have these additional tRNA genes in cloned into their chromosome. So, either of these strategies will overcome the codon usage problem, and it will allow you to overexpress the protein. So, before you decide to overexpress a particular gene, you have to verify as well as you have to test that this problem does not exist, which means you have to critically evaluate the gene structure or the gene sequence to know that the particular type of tRNA would not be a problem. Okay. So, now let us move on. So, the protein which you overexpress in E. coli could be overexpressed as such or the native protein, or you can have the protein which is overexpressed in the form of a fusion protein or the protein which actually going to have the additional tags. So, the protein in E. coli expression system can be expressed as a hybrid protein, which is also called as the fusion protein, where the reading frame of the two gene, one gene which is for your foreign gene as well as the one of the gene which will be for the fusion tag is going to be in frame. So, as a result, when the protein, when the cellular machinery is going to overexpress your the foreign gene, it is also going to overexpress the fusion tag, and as a result what you are going to have is you are going to have the chimeric fusion protein, which means the protein is going to have the additional tag or additional protein from the other portion, and this fusion tag could be placed either on the N terminus, which means it could be placed on this side or it could be placed on the C terminus side. There are many different types of tags which are available for these particular applications. You can have the beta galactosidase as an enzyme as a tag, and the vector what you can use for this purpose is called PUC, P blue script or PGM. What is the advantage of this particular type of tag? You can use this tag for blue white screening as well as you can use the affinity purification. Similarly, you can have the maltose binding protein or the MBP, the vector what you can use where the PMAL is already been cloned is called as the PMAL, and it has an advantage that you can use the PMAL tag or the maltose binding protein as a tag to purify using the affinity chromatography. Similarly, you have the thioredoxin or the TRIX as a fusion tag. The vector what you can use is called PTRIC, and it has an advantage that you can use the thioredoxin tag as in the affinity chromatography. Similarly, you have the polyhistidine tag, and the couple of PET series vectors can be used for, and you have the advantage that it would be used in the affinity chromatography. Then you have the GST tag, the glutathione S transferase tag, and the vector what you can use is the many series of PGX vector, and that has an advantage that you can use that in affinity chromatography or you can use this in the reporter gene assay. Similarly, you have the alkaline phosphatase as a tag, and the vector what you can use to generate the fusion protein is called PTA1529, and that has an advantage to use the alkaline phosphatase in the reporter gene assay. So, what is the advantage of producing the E. coli in the fusion tag? You have the multiple advantages if you are expressing the fusion protein in the form of a fusion tag. One of the prominent advantage is that it actually allows you to do easy purification, which means you see, if you go by the conventional chromatography techniques, you might have to use the different types of chromatography columns like anion exchange column, cation exchange column or the gel filtration column to get the purified protein. And the disadvantage of running the multiple chromatography column is that at every step you are going to lose some amount of protein, and at the end your overall production of that particular purified protein is going to be lower. Compared to this, in the fusion proteins, you are going to use the affinity columns, and these affinity columns are having very exclusive affinity for that particular fusion tag. So, as a result, all other protein when you pass through the lysate to the column, all other proteins are not going to bind to the column, whereas your protein will bind because it has a fusion tag, and then subsequently you will wash the column and elute the protein. So, in that process what will happen is, first thing is you are going to get the purification which is very high. Number 2, you are going to use only single column, which is that affinity column, and that actually will give you very high purification compared to utilizing the multiple conventional chromatography techniques. Number 3, it is going to be fast because you are just simply running the single column. So, it is going to save the time for you. Number 4, since you are saving the time and the protein is getting purified at a faster rate, you are not going to get the protein degradation, which means the protein is going to be saved from the proteases and other enzyme which are present in the cell lysate. Number 5, since you are doing the single step purifications, it is going to be economically viable, as well as it is going to give you the higher production, because you are not going to lose the protein in as you are going to lose when you run the conventional chromatography columns, and in every column steps you are going to lose some protein. And then lastly, since you are going to purify in single step, you does not require a large quantity of human resource to perform the protein purification, which means if you are going to talk about performing the purification in a industrial setup, you need the lesser manpower to purify the protein compared to if you go with the conventional chromatography techniques. Now, since you have the tags, you can actually plan the experiment in such a way that you can actually target your overexpression of your protein to a specific cellular compartment. A fusion protein can be targeted to the different cellular organelles for various regions. So, this is more advantage for the academic purposes. For example, if you want to overexpress your protein to the mitochondria or endoplasmic reticulum or plasma membrane or to some specific sites, you can actually put that particular kind of fusion tag, and as a result what will happen is, the once that fusion tag is present, the machinery will recognize, and that is how the protein will go to that particular site within the cell. In addition to that, in some of the, for example, in the bacterial system, you can actually put the periplasmic localization sequences, and in that case what will happen is, once this fusion protein is going to be overexpressed, the protein will go to the periplasmic fractions. And what is the advantage of sending the protein to the periplasmic fraction is that the periplasmic fractions are away from the cytosolic site. So, isolation of these proteins are going to be easier compared to the cytosolic protein. Number 2, you are not going to face lot of contamination, because the amount of protein which are present in the periplasmic fractions is much less compared to the amount of protein which are present in the cytosol. So, as a result, if you suppose put the periplasmic targeting sequences, the protein will accumulate into the periplasm and hence can help for the easy isolation. Number 3, some of the proteins are having a very short half life, and as you know that the half life of a protein is always been decided by the number of amino acids which are present on the N terminus of the protein. So, some of the proteins are N terminus region is very prone for proteolytic cleavage. In some cases the proteins have its own intrinsic protease activity, and as a result it actually cleaves its own protein as soon as it is synthesized. And sometime what happen is, these kind of thing are problematic because it does not allow you to overexpress the protein in a full length. In those cases what you do is, you produce a fusion protein, and in that fusion protein what will happen is, either depending on what is the scenario of that particular protein, either you put the fusion tag into the N terminus site, or you put the fusion tag on the C terminus site, and either of these sites will be get blocked. So, in some cases what happen is, if you put the fusion tag onto the N terminus site, it actually enhances the overall half life of your chimeric protein, and as a result you are going to have the better survivability of this particular protein or better stability of this particular protein within the cytosol. So, in many cases a fusion tag hides the potential protease site on the foreign protein and enhances its half life. Number 4, the fusion proteins are So, the tag what you are going to put are much more soluble compared to the tag which compared to the protein what you are going to overexpress, because most of these tags are already been optimized that they are being expressing into the E. coli system and they are expressing as a soluble factor. So, once you put a fusion tag and once you generate a chimeric protein containing the fusion tag, the fusion tag component or the counterpart is actually brings the solubility into the overall system, and as a result the chimeric protein is more soluble compared to your own protein. For example, if you put the GST as a fusion tag onto the N terminus, GST is a cytosolic protein, it is very soluble into the E. coli system. So, once you generate a chimeric protein along with this, since the GST is very soluble, it actually gives that solubility parameter to your protein also, and as a result the whole chimeric protein is becomes more soluble. So, keeping the tag at the N terminus direct the protein synthesis and hence help in increasing the solubility of the foreign protein. Now, the question is, once you have the fusion protein, and in some of the applications the fusion proteins are not required. For example, if you are going to use this protein for generating the ELISA, or suppose you want to use this protein for solving the structure using the X-ray, in those cases the fusion tag is going to interfere in downstream applications. For example, if you would like to use this protein which is X for a therapeutic purposes. For example, if you are going to use this X as an insulin. For example, if you are generating a fusion tag of insulin along with the GST, you do not want a patient to receive the GST as well as the fusion tag. In those cases, you have to remove the fusion tag or remove or cut this particular chimeric protein with the help of some proteases, so that the fusion tag is going to be separate. How to do that? So, for removal of fusion tag, for many biotechnology applications, a protein is expressed as a fusion protein either to the N terminus or the C terminus for the easy purification. But after the purifications, the tag need to be removed for downstream applications, such as if we are trying to use this protein for vaccine, for developing the ELISA, or suppose we would like to use this protein for structure solutions, or suppose we would like to use this protein simply for driving catalytic reactions within the patient or into the bioreactors, or sometime if suppose you want to use this protein for therapeutic protein. So, you want this protein to go and inside the cell, inside the human body or inside the host and drive some reactions, and in those cases the fusion tag are going to interfere, or in some cases the fusion tags are definitely going to cause the allergic reactions. In those cases the fusion tag has to be removed. So, for removing the fusion tag, we have the couple of reagents. For example, you can use the cyanogen bromide. Cyanogen bromide is going to cleave the tag just after the methionine. Simply you have the hydroxylamine. Hydroxylamine is going to cleave the fusion tags between the asparagine and proline. Then you have the enterokinase, and you have a enterokinase cleavage site. So, enterokinase cleavage site would be present between the fusion tags, and once you treat this fusion tag with the enterokinase, it is going to cleave here, and as a result the GST is going to be removed and your protein X is free for the for downstream applications. Then you have the factor 10A. The factor 10A cleavage site is also present, and the factor 10A is always cleaving after the arginine. Similarly, you have the alpha thrombin, and alpha thrombin is cleaving the chimeric protein between the arginine and glycine. Then you have the trypsin, and trypsin is cleaving the protein just after the arginine or the lysine, and then lastly you have the subtiliin. Subtiliin is cleaving after the arginine. So, these are not the final list or the exhaustive list of the cleavage reagents which are available for the separating the fusion tag from the foreign gene or foreign protein. Now, the question is how to remove the tag from the your protein. So, for that you can use the same affinity column what you have been using for purifying the protein. So, you can imagine that you have cloned a protein into a his tag. For example, you can use the PET series vector and you have overexpressed the protein. Now, this is your chimeric protein which you have overexpressed, which contains the histidine tags, and now what you do is you take the thrombin, alpha thrombin, and treat it. So, what will happen is the alpha thrombin is going to cleave the linker region between the histidine tag as well as the protein, and as a result you are going to have the tag separately and as well as the protein separately. Now, what you do is take this complete complex mixture and load it onto the affinity column like nickel NTA column. So, as a result what will happen is the nickel NTA is going to bind the histidine tag because it has a very high affinity for histidine tag, but it does not have any affinity for your protein. So, as a result what will happen is the histidine tag is going to bind to the beads of the column, whereas your protein will be present in the into the flow through, and you can collect the protein from the column and you can use this protein for the downstream applications. So, how to do the removal of fusion tag? The fusion tag is in most of the fusion tag you are going to have the protease cutting site, or the site is sensitive for the chemical treatment. So, if you treat the fusion protein with a protease or chemical agent, that will cut the fusion protein to release the target protein. That is what is being done. Now, if you pass this cleavage mixture and allow the binding of the tag to the affinity column, which is actually in this case we have used the nickel NTA column, the column will bind your tag and it will not allow this tag to go into the flow through, whereas your protein has lost the affinity for the column because it does not have the tag now anymore, and the protein will come into the flow through. So, this is all about the overexpressing and production of protein into the E. coli expression system. We have discussed many aspects which are very common for the different for the other expression system as well. Now, we will move on to the next expression system. So, in the list we have the next expression system, which is the yeast as an expression system. So, yeast is the simplest eukaryotic organisms and unicellular eukaryotic organism which you can use for overexpressing those proteins which will not going to overexpress nicely into the prokaryotic system. So, yeast is the simplest unicellular eukaryotic cell available for protein production. It is easy to manipulate and the production cost is very low in comparison to the other prokaryotic system. So, in the eukaryotic system you have the yeast, you have the insect cell lines, you have the mammalian cells, you have the virus or bacteriophage based protein production system. Apart from so in comparison to these systems, yeast is the simplest system in terms of performing the transformations, in terms of the screening the clones, as well as in terms of producing the protein, and ultimately the cost of the protein production in the yeast system is very low compared to the other eukaryotic expression systems. It offers most of the advantages available in a typical eukaryotic cell. In addition, a large amount of genetic, molecular and cellular aspects of yeast is known, and this knowledge has helped us to design better protein production strategies and troubleshooting. So, you know that yeast is very widely studied organisms. So, because of that the yeast is being genetically manipulated. We have very in depth knowledge about the biochemistry of these cells, as well as we know each and every minute details of the molecular biology protocols or molecular biology strategies which you can use to manipulate the yeast as an expression system. And as a result of this, the people are using the yeast very extensively for producing the protein which requires the eukaryotic machinery. Now, in the yeast you have the two choices of host. One is called non-metallotrophic host and the other one is called as the metallotrophic host. Non-metallotrophic host means these yeast molecules or yeast organisms does not depends on or does not accept the methanol as a one carbon source or carbon source, and instead of methanol they use the other carbon sources. So, these species do not have the ability to utilize one carbon compound such as methanol, and that is why these are called as non-metallotrophic yeast strains, but it can be able to utilize other carbon sources such as glucose, lactose, maltose and starch and alkane. The examples in this category are Saccharomyces cerevisiae, Kale lactis and Weil lipolytica. These yeast strains are mostly being used for fermentation to produce the alcohol. The major advantage of this class is better understanding of molecular biology, biochemistry and fermentation technological aspects of these strains. So, the non-metallotrophic host strains or non-metallotrophic yeast strains are not using the methanol as a one carbon source. Instead of that, it is using the other carbon sources such as glucose, lactose and all other kind of sugars as well as the alkane, and mostly these strains are being used for fermentation process. These strains are being used for producing the alcohol and other fermentation related products, and these strains are less popular in terms of utilization, in terms of their utilization for the protein productions. So, these strains are not being utilized in terms of production of heterologous proteins. Now, metallotrophic yeast as the name suggests, these yeast strains are using the methanol as a one carbon source. So, these strains have the advantage that they have the ability to utilize one carbon compound such as methanol as carbon and energy source. In addition, these strains have high level of methanol oxidizing enzyme and that allow them to be very strong and grow in a high density. The example of yeast in this class are Pichia pectoris, Pichia angusta and Pichia methonylica and C. boydini. So, these are the strains, the metallotrophic strains which people very often use for protein productions. Now, if you want to use the yeast as an expression system, the first step is that you are going to transform the recombinant DNA what you have generated into the yeast vectors. So, we have discussed a couple of yeast vectors in the past, and we have also discussed about the transformation methods where you are going to use either the spiroplast based transformation methods or the lithium acetate based transformation methods. So, for protein production mostly people use the lithium acetate or the electroporation as a popular method for transformation of yeast. The transformations are then selected based on the kind of vector what you are going to use. So, in the second step you are going to select the vectors as well as the transformed host strains. Either you will so mostly people use the auxotrophic markers such as URA3, leucine 2 or tryptophan 1 or histidine 4, or sometimes you use the antibiotic resistant genes such as the G418, hygromycin, etc. Once these two steps are over, then you have to use the promoters. So, just like as we discussed in the E. coli expression system, there are different types of promoter also present in the yeast as an expression system. So, you have the two different types of promoter present in the yeast expression system, either you have the constitutive promoters or the inducible promoters. So, in the constitutive promoters, these constitutive promoters are the promoter which are present in the housekeeping genes. So, these promoters actually belongs to the housekeeping gene, and as a result the expression is non-inducible, which means they are going to give you the protein, but that protein production would be in proportion to the number of cells what you are going to have. You are not going to be able to use any inducible agent. For example, if you remember in the case of E. coli, we were using the IPTG or lactose or some kind of inducible system, where we were using that kind of environmental changes. For example, when we were using the CI3 promoters, we were changing the temperature, and as a result changing the temperature, it is actually also driving the protein production. But in these cases you cannot do that, because most of the constitutive promoters are the promoter which are belonging to the housekeeping genes, and you know the purpose of housekeeping gene is to produce the protein at a consistent rate and throughout the life of the particular organisms. One of the classical example is the LDH. So, LDH protein is a housekeeping gene, or actin or myosin. Most of these structural proteins which we are present, or which are actually be a part of the formation of the plasma membrane, are actually belonging to the housekeeping genes. So, housekeeping genes are never been inducible, because they are being consistently producing the protein. The protein production starts with the growth of the yeast, and as a result it is proportional to the cell mass. Example of these promoters are GAP-DPH, GM1, etc. Now, the second class is the inducible promoters. Inducible promoters are the promoter where you can actually use some class of inducers to produce the protein in a large quantities. Examples are Pichia pectoris expresses two alcohol oxidases like OX1 and OX2, whereas the Pichia augusta expresses methanol oxidase MOX, and all these three genes, if you take the promoter out of these three genes, these promoters are going to be inducible promoter, because as soon as you add the alcohol, these genes are being the promoter which is present in front of these genes are going to generate the large quantity of these proteins. The promoter of OX1 and MOX are present on yeast vector, and it has been used to drive the expression of a foreign protein in the inducible matter. The protein production is controlled by a balance of repression and induction. Presence of other carbon sources such as glucose represses the transcription of OX1 gene, but in the presence of trace amount of methanol, it induces the OX1 promoter mediated protein production. So, inducible promoter, just like if you remember, if you have the very small quantities of lactose, that actually is going to give the leaky expression in the E. coli system. Similarly, here if you have the alternate carbon source, for example, if you have the glucose, the yeast is not going to use the methanol as a carbon source, instead it will use the glucose as a carbon source. In those cases, the glucose is going to repress the activity of OX1 promoters, but if you use the methanol or tiny amount of methanol, and that actually is going to induce the OX1 promoters, and as a result you are going to see the protein production if you have cloned the protein in front of the OX1 promoters. So, this is just a summary of different promoter as well as the expression systems present in the E. coli expression system. You have non-metholic strains such as S. cerevisiae, K. lactis or Y. lipolytica, whereas you have the methylononic strain such as Pichia pectoris or Pichia methanolica. You have the constitutive promoters such as gap DPH and all other kind of promoters, whereas you have the inducible promoters such as ADH1, ADH4 and OX1, MOX, and all these promoter can be used to drive the protein production into the yeast as an expression system. You have the choice that you can produce the protein either into the cytosol, or you can have the choice that you secrete the protein into the external supernatant, and that depends whether you have the any kind of tag which is present on the protein. So, if you have a tag which is present on the protein, that protein will going to secrete out. So, cytoplasmic targeted proteins, the expression of the protein targeted to the cytoplasm is very high, but the recovery is very difficult. Yeast cell is very hard and high pressure homogenization is required, homogenizer is required to disrupt the cell wall. The recovery is very less and a fraction of total soluble protein comes out. So, if you overexpress the protein as a cytosolic protein, the protein production is going to be very high, but the disadvantage is that since the E. coli the yeast has a very strong cell wall, the cell wall is not going to be broken very easily, and as a result, even if you use the high pressure homogenizers like French press and all other kind of homogenizers, even then the recovery of the protein from the yeast cell is going to be very less. Now, if you are putting secretory components like protein, if you protein tagged with a secretory signal such as S cerevisiae, alpha mating factors, that actually will promote the secretion of the protein into the secretory pathway, and that actually the signal peptide is processed in E. coli vesicular transport system and appeared in culture media. So, in that case the protein will be secreted out, it will present in the culture media, but it depends about the protein which is going to be overexpressed. It is very difficult to decide whether you would like to use the cytoplasmic pathway or the secretory pathway, because both are these pathways have their own advantages as well as the disadvantages, and that actually you have to decide depending on the type of protein which you would like to overexpress in yeast expression systems. Now, we have the multiple steps which you have to follow to for the protein productions in the yeast expression system. The first step is that you do the transformation of the yeast with the recombinant recombinant DNA. So, transform once that is the once you generated the transformed yeast, the first step is the transfer the transformed yeast into a 5 ml media with suitable selection markers and incubate that for 2 days at 28 degree Celsius with shaking at 180 rpm. So, the first step is that you first transform the bacteria, first transform the yeast with the foreign DNA or recombinant DNA, then inoculate that into a 5 ml media with selection marker and then incubate that on 28 degree Celsius for 180 rpm. That actually will allow the yeast to grow at a very high density. Once it reach to a specific density, then you allow the culture to reach with the OD of 5 to 7, and now resuspend the cells in a new media without carbon source. So, in this particular type of media you are not going to use the methanol as a carbon source. Here you are going to use the regular carbon source such as glucose or other kind of carbon sources, and then you allow the yeast to grow to a OD of 5 to 6. Once it reaches to a OD of 5 to 6, then what you do is you pellet down these yeast cells, put it into a new media, and in this new media you are not going to provide the glucose or other 1 carbon sources as a for the carbon source. Instead, you were going to induce the culture with the methanol, the 1% methanol twice daily, and as a result what will happen is since you know that the methanol is very toxic, but for these particular type of strains, because they have the ability to metabolize the methanol, you have to use the very small quantity of methanol, only the 1% methanol you have to use, that too twice daily, and that will actually induce the promoters of OX1 or MOX, and that actually will allow the production of the protein. Once the protein is being produced for in couple of days, because that actually will take time, then you harvest these cells by centrifugation and analyze the expression on the SDS page, just like as we discussed in the E. coli system as well. Now, so far we have discussed about the E. coli as an expression system, we have discussed about the yeast as an expression system, and now we will move on to discuss about the insect cell as an expression system. If you would like to use the insects as an expression system, you have to use the what you have to do is you have to perform the multiple steps to achieve the protein production in the insect cell lines. So, as a eukaryote, Biochemical expression system offers protein modifications, processing and transport system. Compared to yeast, the downstream processing and recovery of cytosolic protein is much easier in bacterial expression system. If you remember, we have discussed that the yeast has a very strong cell wall, and because of that the recovery as well as the processing of these cells are much more difficult, which is actually going to be simplified in the bacterial expression system or the insect cell line as an expression system, because insect cell lines are easy to break and the recovery is much better. So, if you would like to produce the protein, you have to follow these steps. First, you have to clone the foreign gene into the transfer vector, then you have to generate the recombinant baculoviruses or baculovectors, then you have to screen the recombinant baculovirus, then you have to culture the recombinant insect cell lines, and ultimately you have to do the protein production. So, let us start with the first step, and the first step is the cloning of foreign gene in transfer vector. We have already discussed about the design of the vectors which are available in the baculo expression system. In the baculo expression system, you have a vector which is actually containing the flanking sequences which are present from the viruses. So, you have the N terminus flanking sequences, you have the C terminus flanking sequences. In between, you are going to have the promoter, which is for the polyhedrin promoter, a polyhedrin promoter. Then you have in the middle of in front of the promoter you have the cloning site, and then next to the cloning site you have the polyhedrin termination site. So, that this polyhedrin promoter is going to drive the production of the gene which you are going to clone into the cloning site, and the upstream as well as the downstream sequences, which means these sequences are required are from the virus genome, and that actually helps in homologous recombinations. So, what you have to do is for the cloning of the foreign DNA into the transfer vector, you clone your particular protein into the cloning site. Now, the next step is the generation of recombinant baculovirus. You have the two approaches to generate the recombinant baculoviruses. In approach 1, what you are going to do is, you can imagine that you have the insect cell lines. So, first in the first step what you do is you take the insect cell line and first transfect with the plain baculovirus, and that actually will going to produce the transformed insect cell line. Now, in the second step what you do is you transfect these cells with a transfer vector containing the foreign DNA. Now, what will happen is once these two vectors, the transformed insect cell lines where you have already put the viruses, and these transformed cell when they will be transfected with the transfer DNA containing the foreign gene, once they will replicate for 2 to 3 cycles, what will happen is there will be a homologous recombination between these two factors, these two regions, which means the upstream as well as the downstream region are going to do the homologous recombination. So, this is the transfer vector which you have transformed in the second step, and this is your plain virus. So, as a result what will happen is this whole cassette from this place is going to be replaced with your foreign gene, and as a result you are going to generate the recombinant DNA or recombinant virus, which actually will going to contain your gene or your cassette instead of the virus its intrinsic polyhedrin gene. And as a result, now what you do is you take this virus and you can use that for protein production. In the approach 2, what you have to do is, in approach 2, the baculovirus genome is engineered to introduce two unique restriction site BSU 361 onto the gene 603 as well as onto the gene 1629, and both of these genes are present in the present in the polyhedrin gene within the viral genome. When modified viral genome is treated with the restriction enzyme, so when if you take this modified baculovirus, treat it with the BSU 361, what will happen is it is going to be cleaved off from here, and you, as a result you are going to remove the complete cassette of polyhedrin gene. So, you are going to lose this particular fragment. Now, once you if you transform this particular fragment, the cleaved fragment, you are not going to get any viruses, because you are actually missing the crucial function of the gene 1629, which is required for the viral replications. Now, this linearized viral genome or linearized truncated viral genome with missing polyhedrin gene ORF 1629 and the gene 603 is transfected into the insect cell, followed by a transfection of transfer vector containing the foreign DNA along with the gene 603 and essential gene in one or two division. So, once you take this particular gene and transfect into the insect cell lines, once they will go with the 1 or 2 cycles, this particular region is going to go with the homologous recombinations, and as a result the gene, this particular cassette is going to be replaced with this cassette, and you are going to produce the recombinant baculovirus, where you are going to have the gene 603 in the front and gene 1629 on the back side, and in between you are going to have the gene of interest cloned within the within the promoter of polyhedrin gene. And now this recombinant baculovirus is ready for protein productions. So, a double cross event will occur. A double cross event will occur between the truncated viral genome and the transfer vector with the help of flanking gene 603 and essential gene sequences. As a result, the viral genome receives the lost portion of gene 603 and ORF 1629 from the transfer vector, and the foreign DNA is incorporated into the viral genome. So, as a result you are going to generate the recombinant baculovirus, recombinant baculovirus. So, now whether you use the approach 1 or whether you use the approach 2, you have generated the recombinant baculovirus. Now, these baculoviruses can be used for protein production. Now, the subsequent step is the screening of these baculoviruses. The recombinant baculoviruses can be screened by a plaque assay. The major steps in the plaque assay is that first you dilute the culture of insect cell line SF9 to a density of 10 to the power 5, make serial dilution of baculovirus stock in serum containing media, add 1 ml of virus sample to each and incubate for 1 hour at 27 degrees Celsius. Now, remove the viral diluted suspension and then detect the presence of plaque. There are 3 methods, 3 popular methods to detect the plaques. What are these methods? You overlay the agarose and allow it to harden. Incubate this plate for 6 to 8 days at 27 degrees Celsius to plaque form, and these can be visualized. Number 2, if the recombinant virus contain a LAGZ gene, for example, then the plaque can be identified by simply adding the beta galactosidase substrate such as XGAL, and XGAL is going to give you the blue color. So, the plaque containing cells will appear the blue. In third method, cells can be stained with a trypan blue, and plaque containing cells will take up the dye and appear blue, whereas the other cells will remain colorless. So, plaque is nothing but the holes within the cell. So, once you add the trypan blue, the trypan blue will directly go into these cells and it will give you the blue color. Now, once you are done with the screening, you can use the these recombinant baculoviruses for protein productions. Before you go into the details of protein production, you have to do the culture media for the growth. We have discussed different culture media in a previous lecture, and which you can use to grow the insect cell lines. So, insect cell line SF9 is being derived from the ovaries of the armyworm, and it is maintained in TNH FH insect media containing 10% FBS and gentamicin. For protein production, you can use either the baculo gold or other serum free media, because if you keep the serum in the protein production media, and if the protein is secretory in nature, you are going to have the high level of cross contamination or high level of contamination from the serum protein itself. Low protein media is suitable for the secretory protein as it facilitates the easy purifications. Now, let us see what are the different steps we have. So, the whole process of protein production is as such. You first, what you do is you seed the 10 to power 6 SF9 cells in a 60 mm cell culture dish and allow the cells to adhere to the dishes. Now, add 0.1 ml high titer baculovirus stock at a MOI of 1 to 10. Now, incubate the cells for 3 days at 27 degrees Celsius, so that the virus can infect the insect cells and it can propagate within the insect cell. Now, collect the cells and media, centrifuge at 1000 G for 10 minutes at 4 degree. So, the first step, you do plating in a 60 mm dishes and allow the cells to adhere, and then you make the single layer. Then you do the transfection with the help of the baculovirus at the MOE of 1:10. Once these cells are being transfected, then what you do is you culture them and collect the supernatant as well as the cells. Now you have the options. If the protein is secretory in nature, which means the protein is going to be present in the cell culture. So, you transfer the culture supernatant to a new tube and determine the protein concentration with a Bradford reagent, which means if your protein is secretory in nature, you can just collect the culture supernatant and you can estimate the amount of protein present. If the protein is cytosolic in nature, then you can recover the insect cells, you lyse these cells by using the different lysis method, and that actually is going to give you the cytosolic protein. So, if the protein is cytosolic in nature, discard the supernatant and wash the cells pellet with PBS, then lyse the cells and analyze the protein on the SDS page. So, with this, these are the different steps which you have to follow to overexpress the protein into the baculo expression systems. So far what we have discussed about the E. coli as an expression system, we have also discussed the yeast as an expression system, and lastly we have discussed about the insect cell line as an expression system. We have discussed about the different approaches and different options what are available in all these different expression system, and Now, in a subsequent lecture, we are going to discuss about the mammalian cells as an expression system, and with this I would like to conclude our lecture here, and in our subsequent lecture we are going to discuss about the mammalian cells as an expression system. Thank you.